Before taking a look at recombinant vaccines, let's look back into the history of vaccines.
Going back 1,000 years, a viral disease known as smallpox started to affect many people around the world, causing fever and permanent scars leading to death.
Physicians first practiced immunization of smallpox using a method called variolation. They took scabs of patients affected by smallpox and crushed the scabs into powder. The powder was then blown into the noses of patients, which resulted in the patient to become immune to the disease.
Several years later, in 1798, a doctor in England named Edward Jenner discovered the smallpox could be eradicated using the cowpox virus as immunization.
In 1877, Louis Pasteur proposed the germ theory of disease. He then invented the first attenuated viral vaccine in 1884, which was then tested on humans in 1885.
By the 1930s, the study of the behavior of single cells was advanced to a stage where it was discovered that viruses were able to grow in the cell. The production of viral vaccines were then highly mandatory.
Vaccines are weaker versions of bacteria or a disease, which are then injected into the body so our immune systems are able to analyze and familiarize themselves with the disease before actually coming in contact with the actual disease. When an immune cell comes in contact with the actual disease, it duplicates itself in order to produce antibodies which eliminate the disease. Once the infection is destroyed, the only thing that remains are what we call memory cells that are ready to attack if the infection is seen again.
Many social groups have protested against vaccines, starting from the immunization of smallpox. People believe that the use of cowpox would actually turn people into cows.
Other people believe vaccines led to autism due to the toxins they contain. The studies show that there was no connection between autism and vaccines. In 2009, the U.S. Court of Federal Claims confirmed these studies.
In the year 1976, an outbreak of the swine flu that started in New Jersey became a huge concern for everyone, including doctors, scientists, and researchers. Scientist Maurice Hilleman and other scientists got together and discussed with the president a vaccine response for the swine flu outbreak. Maurice anticipated to create 50 million vaccinations for the swine flu by the beginning of 1977 and ship out about 11 million doses of the vaccine in September of 1976. Unfortunately, the nationwide vaccination program ended because it was found that the vaccine was linked with a bigger risk of a certain condition called GBS syndrome.
In 1981, the first hepatitis B viral vaccine, which was developed by Merck and the Pasteur Institute, was licensed.
In 1985, the hepatitis B recombinant vaccine was tested using 200 healthy men. In the end, 95% of them were protected.
By 1986, the first recombinant hepatitis B vaccine developed by Merck scientists was licensed. The number of countries that had access to this vaccine increased from 31 in 1992 to 164 by 2006.
In 1988, recombinant vaccines were promising against the dengue fever.
What exactly are recombinant vaccines?
Recombinant vaccines are made using recombinant DNA technology and consist of protein antigens, which are produced from the heterologous organisms such as yeast or bacteria. Once this antigen-containing vaccine is injected into the individual, the antigens produce antibodies to prevent and protect the body from diseases.
Other examples of recombinant vaccines are hepatitis B, chlamydia, diphtheria, and tetanus.
Hepatitis B recombinant vaccines are produced as a result of inserting a hepatitis B virus into the gene of a yeast cell. As the resulting yeast cell grows, it produces a hepatitis B surface antigen.
Scientists pursue this experiment because it does not consist of any bacteria or other organisms that can cause diseases, so they are considered safer than general vaccines.
The vaccination is also much more effective compared to a DNA vaccine due to a greater immune response.
Another advantage of recombinant vaccines is it mimics an infection closely, allowing the immune system to adapt and build a stronger defense system.
Many individuals do not consider the seriousness of getting your vaccine shots. If not vaccinated, the individual is more prone to get the disease and spread it to those around them. This was seen with measles in the year 2000 when the CDC announced they were destroyed. However, some people failed to get their measles vaccine, and so by 2011, 220 Americans were infected with measles.
Although there are many benefits of recombinant vaccines, there are still some flaws.
First of all, the manufacturing process is costly.
Along with this, the vaccine has to be stored in a cool environment because if not, the heat may damage the protein, meaning the storage and shipping of these vaccines require a lot of work and time.
Individuals whose immune system fails may bring forth a poor response from the immune system when a vaccine is inserted into the body. 2.
Scientists also have to think about those with yeast allergies. Since recombinant vaccines are made from yeast, some individuals may be allergic to the yeast and could have an allergic reaction to it.
Researchers are working very hard to make the world a healthier place by developing vaccines to fight against malaria and HIV, as well as working harder to make cheaper vaccines that can be shipped out to countries with poor economies. Researchers are continuing to work on delivery systems that can deliver the vaccines more efficiently so people can receive them quicker.
We are yet to discover several other bacteria and diseases in the future that are harmful to humanity that may just be right around the corner. With the help of education and research, a proper vaccine can be developed to eradicate these diseases that we may face. We are all in this together.